So today we're going to deal with the last system of the body, and this is the urinary system. So this is responsible for the production, the storage, and the elimination of urine. Now, urine is first produced by the kidneys, which are two organs, kidneys, and they're stored. The urine produced is stored in the bladder via the ureters, and then it's ejected in the urethra. Now, only a small fraction of blood plasma processed by the kidneys every day is secreted as urine. So you should know that it's basically a quarter of the cardiac output is going to be the kidney urine secretion. And remember, cardiac output is liters per minute. And this would already also be the urine secretion is producing this much liquid per minute. And urination relies on the coordination of the bladder and urethral sphincter muscles through the autonomic and somatic nervous system. And two urethral sphincters must relax and the detrusor must contract. So let's write this down first. Is the detrusor contract. And the first one that needs to relax is the internal urethral sphincter. And the second one is the external urethral sphincter. An external urethral sphincter is through somatic. And the internal is by autonomic nervous system. So I guess this is kind of the first idea. You need like the, give me a second. You need to, and this is also by the autonomic nervous system or the detrusor. Yes. So yeah, you would have to inhibit the detrusor contraction through autonomic ganglia. Oh well, you would have to contract the detrusor, so we have sympathetic activity, and you would also need to have relaxation of the urethral sphincter, so parasympathetic activity on the urethral sphincter, and then also somatic inhibition onto the external urethral sphincter. So the kidney tissue is divided by the cortex and the medulla. And they have many functions. One is removing wastes and toxins, blood. Two is regulating ion concentrations. Three is regulating blood pH. Fourth is regulating blood volume and pressure. It's also a secondary endocrine organ and is a target of endocrine organs. This is a target of aldosterone, ADH, AMP, and PTH. So the first one, hormone 1, what we can talk about is called calcitriol. Calcitriol regulates calcium intake or retention. So first, vitamin D is either from the diet or from the skin, enters through the liver, which turns it into an intermediate product. And this intermediate product goes into the kidneys, which turns it into calcitriol when calcitriol when responding to PTH signaling. And this is due to low CO2 levels. And then due to calcitriol, it stimulates onto intestinal receptors in the digestive tract. And it also stimulates onto kidneys, called absorption of calcium. This increases blood calcium concentrations. The other thing is EPO. Oh, and I should mention, this is secreted from the calcitriol is secreted from the proximal tubular cells. Everything is EPO or erythropoietin. EPO is secreted by intertubular fibroblasts. And this is during hypoxia. It acts upon red bone marrow to produce red blood cells, leading to increase in blood volume. Third one is renin. Renin is secreted by cells of the juxtaglomerular apparatus. Big word, juxtaglomerular apparatus, JGA, and is secreted in the bloodstream. This is due to low pressure in arterioles. I guess you say low blood pressure, bloodstream. And then liver, in this case, liver secretes angiotensinogen, which renin in the bloodstream cleaves angiotensinogen to angiotensinogen 1. Angiotensinogen 1 into the lungs secrete ACE, which is angiotensin and angiotensin converting hormone enzyme, which converts it into angiotensinogen 2. Angiotensinogen 2 can act onto the adrenal glands to secrete aldosterone. This is for water reabsorption. Angiotensin 2 can also directly lead to vasoconstriction. And basically, water reabsorption and vasoconstriction all lead to increases in blood pressure. The functional unit of the kidney tissue is nephron. So first has a glomeruli, glomerulus, and then you have a renal tubule. So you have a glomerulus, a renal tubule here. This is where it's filtrated. After filtration of the glomerular capillaries, the filtrates enters the nephron, and this begins filtration. So remember, filtration at glomerular capillaries before going to the nephron. And remember, filtration is moving fluid through small pores due to pressure differences. And yeah, interactions between renal tubules, interstitial fluids shape the composition of urine. So you first have cortical nephrons, which are near the cortex and renal cortex. And then you have the juxtamedullary nephrons, just close to the medulla. And cautery nephrons are around 85%, where juxtamedullary are 15%. And because they're closer, the juxtamedullary have an extended, they have more reabsorption of water and solutes. This is because they have a more extended renal tubule. Longer renal tubule. And the composition of urine is variable because a lot of it is due to hormonal regulation. So what's important to know is obligatory processes happen in the proximal tubule. So if we were to draw it here, the glomerulus it will look like this. So this section is obligatory. So it'll be the glomerulus, proximal convoluted tubules, and then the descending duct, descending limb of nephron. And then this would be ascending limb of nephron, distal convoluted tubule, and collecting duct. And this is facultative. So not always present. Renal failure is when renal function is compromised. So acute is abrupt loss, whereas chronic is gradual deterioration. And this can be caused by many things. One is clot, renal vein or artery. Two is physical damage. Three is toxins breaking down. And four is stones blocking a ureter. This leads to inflammation, vascular injury, and tubular death. And kidney failure can be managed using hemodialysis. So hemodialysis is basically a machine that, let's say you're hooked up to, and your blood is pumped in, and the blood just kind of inserts the dialysis fluid. So blood goes into dialysis fluid, and it's filtered and then returned back to the body. And this generally takes 3 to 4 hours, 3 times a week. And dialysis, it relies on passive diffusion through permits and restricts large molecules. So it uses a passive process, especially plasma proteins. And this is designed to prevent the loss of nutrients and key ions from plasma. So one thing you might want to understand is there might not be something else they show you where it's like, something here, and you have what it is in plasma and dialysis fluid. So let's say we have anything, if we have more something in the plasma and less in the dialysis fluid, the solute is going to move out due to the fusion because we're only like a passive processes. So if it's something important, like let's say glucose, you don't want glucose to be eaten out. So you want to have a high concentration of glucose in the dialysis fluid relative to a lower one in the plasma. Let's say it's a waste product, like let's say we want to look at toxins, then we will want to have a high concentration of the plasma and basically none in the dialysis fluid, if that makes sense. So these sort of things have to be regulated on a case-by-case basis for patients and all that. Now let's talk about glomerular filtration. Glomerular filtration. Filtration happens in the glomerulus. And there's 3 processes that basically happen in neurons. One is filtration, 2 is reabsorption, and 3 is secretion. Reabsorption and secretion occurs in the renal tubules and the collecting. So in the glomerulus, in the initial portions, they first have a cortex. And yeah, the glomerulus is the blood vessels between afferent and efferent arterioles. They have a capsule, and well, I should say it's not like a cortex, I should call it a capsule, which encapsulates the glomerulus, and the glomerulus contains blood vessels between the afferent and efferent arterioles. So what it looks like is, let's imagine we have an afferent arteriole going in and an efferent arteriole here, and then the blood vessels between them are what's called the glomerulus, and then we have a renal capsule. This entire system is called a corpuscle. And yeah, They also have something called a juxtaglomerulus apparatus. So the juxtaglomerulus apparatus. Remember how we mentioned it's the EPO secreted from here? Okay. Oh no, it's renin that's secreted from here. EPO is actually, just as a quick reminder, I suppose, EPO is secreted from intertubular fibroblasts, and calcitriol is secreted through the proximal tubule cell. Proximal tubule cell. Okay. And the juxtaglomerular apparatus is here, and this detects changes in arterial pressure and flow filtrate. Afferent arteriole has a larger diameter, efferent has a smaller diameter. And you have a distal convoluted tubule, and then you have the proximal convoluted tubule. The main idea is you have a visceral layer of the capsule, and which contributes to filtration, but you also have a capsular layer, which has space that contains a filtrate. So I'll label it capsular layer, which is space, and then the visceral layer. This leads to filtration. And the filtration membrane is made with these endothelial cells, basement membrane, and it also has these things called podocytes. The podocytes, well, they're first fenestrated. They have fenestrated sinusoidal endotheliums. And the basement membrane prevents movement of large molecules. But they have these podocytes, and podocytes have feet which wrap around the basement membrane. Basically, you can imagine they look like this. And with these slits, you can allow proteins to leave in and out. Well, not proteins, but smaller molecules. So we have podocytes with feet, and they leave in filtration slits, allowing small molecules to move. So what can be filtered then? It's any molecules less than 10K Dalton can move through without obstruction. If it's between 7K to 10K, sorry, less than 70. Oh, it's between 70K, sorry, to 10K 10K Daltons. Then it can still move, but less efficient. And they'll either be reabsorbed or pass in urine. And then if we're 70 Dalton, not possible to pass through. This is just part of glomerular filtration. And by making the filtrate, we have a glomerular filtration rate, which is usually a quarter of the cardiac output in a day. This is measured in milliliter per minute. And now remember how we talked about in the blood we have pressures that move fluid in and out. The same thing happens in the glomerulus. So if we imagine the glomerulus here, then basically the glomerular space or the capsular space, you first have a pressure fluid that's in the glomerulus. It wants to move out. There's also proteins wanting to move in, so let's write this way. So the net filtration pressure, first if we're talking only about the glomerulus, it's going to be 100% pressure in the glomerulus. So we can call that GHP. And then this is going to be subtracted by the proteins making it want to go back in. So this is G No, it's still BCOP because we're filtering blood. This is going to be BCOP still, and then GHP. At the same time, there's going to be stuff inside the capsular pressure, and the capsular pressure is going to actually want to move in, so that's going to be adding CSHP, and there's also going to be some proteins capsular, maybe, and this is going to be plus CSCOP, capsular COP, but minus capsular COP. But usually capsular COP is actually 0 because there should be no proteins in the capsular of the glomerulus. This is because proteins can't pass, remember, with the podocytes making only small entrances for small molecules. So what's going to be is it's going to be the GHP plus the CSHP minus BCOP. Those are the players. And this can basically alter this can also be altered in different ways. So if we constrict afferent arteriole, so arteriole here is leaving it, we constrict the afferent arteriole here. This means it'll be harder for fluid to leave out, and this would increase. So GHP would increase. Why? Because we're not talking about capsule. The capsule is irrelevant for the arterioles, but it will increase because, well, the blood has to stay here more. It's going to be filtered more, and there's going to be more hydrostatic pressure. And then if we block drainage, so let's say we got block drainage of urine from the nephron. So this is going to be, the drainage is going to go this way. Then it's going to, if we're going to prevent this, then we're also going to increase CSHP in this case, right? And yeah, the main sensors for the homeostatic regulation of GFP are found within the juxtaglomerular apparatus. And this is due to, well, this is due to mechano and chemosensory cells. So granular cells responsible for mechanical sensation via blood pressure, via the arterial stretch. So this is mechanical, and then the chemical is part of macula and hence the composition and flow rate of filtrate within nephron. And basically the granular cells, they look at arterial stretch in order to measure blood pressure. And then the macula tensa senses the composition and flow rate, which leads to other signals. And the main effectors of the homeostatic regulation GFRP, I mean GFR, are arterial smooth muscle within the glomerulus. So they'll lead to contraction of, let's say, smooth muscle and list out in the arterioles for one. So the afferent, and then the intraglomerular mesangial cells. And this is due to the tunica media layer constricts arterioles. GFR is also regulated by intrinsic mechanics. For example, this can also lead to myogenic reflexes, where if we have an increase in arterial pressure, arteries are stretched, then smooth muscle reacts by contracting. And in doing so, this increases resistance. And then disruption is also sensed by JGA. And let's say if there's a decrease in renal blood flow, and JGA leads to paracrine signaling. Or in, let's say, the case of renin, there's also its extrinsic regulator through the endocrine and hormonal systems. And this is done through the macula tensa and the granulocellular cells secreting renin. And then, yeah, renal afferent neurons can enter the CNS at the thoracic spinal cord. Renal afferent neurons, thoracic spinal cord. And that's how it is. This leads to intrinsic signals. Extrinsic signals alter GFR through the effects on the cardiovascular system. And there's also ways that we might want to understand on how to measure GFR empirically. So the question is, how do we estimate GFR? You can't stick a sensor in individual glomerulus, so we can look at a substance that's filtered by kidneys but not secreted or reabsorbed. One way is using estimated GFR, taken from plasma levels of key waste products. So if you guys think of a waste product that could be secreted, I'll be back in a bit. Think about it. If not, the answer was creatinine. So creatinine is from creatine, which composes to decomposing the creatinine, and this is creatine is found in muscle, and this could only be excreted via kidney. It's not filtered, not reabsorbed, and it's not secreted in the renal in the kidney or in the body in general. So because it's only excreted, if we measure the amount of creatinine in blood, you can give a portion of prediction of GFR. So measurements in blood estimates GFR. So for example, if someone has high creatinine, that means that their filtration might be kind of low. Well, high creatinine in blood, that means it might be low. Their filtration might be low because they're keeping too much of it. They have low creatinine, then it might be high. Then the filtration is high. There's also matters of diet, but you can see a graph of plasma creatinine versus filtration rate. If you have low creatinine, you're going to have a low filtration rate. Another way is you can directly measure it. This is determining the rate of clearance of inert substances. And for example, they make you drink drug, use a drug with particular properties, and then see it in urine. One example of this is inulin. It's non-toxic, not metabolized, only excreted by kidneys, filtered, not reabsorbed. This is injected onto an IV, maintain steady plasma concentration. Then for urine collection over hours. And the amount, you calculate the concentration times the volume, the amount of the recovered in a line. You can measure that and then determine basically how much is being filtered. Now let's talk about the tubular reabsorption and secretion. So we talked about more of the glomerulus, but one case we can actually review on is actually the idea of diabetes. Diabetes is increased glucose metabolism. We have increased glucose levels in the blood plasma, and this leads to two things. You see urinary glucose in the urine, and you have more urine production. This is what's detected. And yeah, remember how the three basic functions were filtration, reabsorption, and secretion? Now we're talking about reabsorption and secretion. So the renal tubules plus the collecting duct have three basic functions. Remove nutrients and proteins from fluid. 2, reabsorb filtered ions. And 3, concentrate waste products in tubular fluid. So PCT is most of the reabsorption and a bit of secretion with a nephron. And they absorb basically glucose, sugars, amino acids, and then secrete hydrogen, creatinine, drugs, and uric acid. And PCT is only part of nephron that can absorb organic substances, such as nutrients or filtered protein. So this is a key idea. PCT is a leaky, simple cuboidal and has microvilli. So this allows for paracellular transport. And then microvilli leads to increased surface area, allowing for transcellular transport. Reabsorption also occurs along the proximal convoluted tubule, and the co-transports are found on the apical side. So the co-transports, apical side, guess we should say with an apical basolateral. So this is going to be the lumen. So the apical side, the co-transporters are going to basically for Na for glucose or Na for amino acids. And basolateral reflects transporters for glucose AAs. Basolateral side also have NAKTases for gradients. Co-transport allows for 99 to be filtered, 99% to be removed through this form of transcellular pathway. And then this is returned to the paratubular capillaries via diffusion And reabsorption of water also happens. Water, and this is through osmosis. This is also through the cellular pathway. This is around 2/3 of filtered fluid per day. If glucose are pumped out of the lumen into PCT cells, the osmolarity of interstitial fluid will decrease, and the osmolarity will also, well, yeah, it'll decrease, but SCF and ISF will increase. The osmolar gradient will draw water through the ISF into the tight junctions of the epithelium. In diabetes mellitus, so you can see so I should draw it like this. So if the osmolarity of turbule, if more glucose is pumped out into PCT cells, then the osmolarity, let me check for a sec actually. The osmolarity of tubular fluid will decrease and the turbulent fluid will decrease. So meaning we'll see essentially less solids will decrease osmolarity, but the osmolarity here will increase. So more solids. And in addition, so water will draw water into the ISF with leaky tight junctions. So let's draw the steps first. When solids enter the ISF, the tubular fluid osmolarity decreases while ISF osmolarity increases. This gradient, I should say osmotic gradient, pulls water into ISF via leaky tight junctions of the epithelium. Extra filtered. So if you have extra filtered glucose, meaning if you have filtered glucose that isn't entering ISF, that leads to excess urine production because there's going to be water staying. And if plasma concentrations of glucose are high, in that case, glucose transport will saturate. Remember, transport will saturate if glucose is too high. The plasma. And if glucose is not reabsorbed, the water will remain. So what happens is osmolarity of tubular fluid remains high. Osmotic gradient of ISF, say from lumen to ISF, is reduced, and then less water reabsorbed by PCT epithelium. More remain in lumen. And this leads to symptoms of glucose in urine and increased urination. PCT cells also absorb albumin and other proteins via endocytosis, or I should say receptor-mediated endocytosis, and they're returned to the plasma or broken down, excreted. The proximal convoluted tubules have functions beyond organic compound absorption, and well, PCT and DCT. They also secrete nitrogenous wastes, metabolic acids, and toxins or drugs. So by secreting acids, they regulate pH this way. and epithelial cells. So epithelial cells in PCT and DCT have carbonic anhydrase. This converts CO2 to H2CO3, later to HCO3 minus plus H plus. And then this is excreted via counter transporters. And yeah, once in a lumen, this can reform and whatnot. Once in a lumen, that's being secreted. Because remember, trying to get the good stuff, whereas getting rid of the wastes. So once in a lumen, H plus can reform as HCO3 and allow for simply just HCO3 reabsorption. Can reform for absorption. And then this secretes the H plus. This is done through a sodium hydrogen countertransporter and a H countertransporter. And then we also have an NA pump, TPA's pump, I should say. And then they both secrete nitrogenous wastes. One thing you should know is NH plus is by counter transport, while NH3 diffusion in plasma membrane. And the drugs, we have drugs, toxins. This is the organic, general organic cation anion transporters. They transport a whole variety of organic and just just general compounds. They express cells for that. And yeah, there's a lot of drugs that can cause damage to the kidneys when taken in high doses, and they're present when kidney function is reduced. And also toxicity occurs because the cells must concentrate to process these drugs or to eliminate, which definitely causes strain and damage to the kidneys. And I guess the last part of the kidney lecture is going to be the regulation of urine and the creation of the medullary osmotic gradient. So each segment of nephrons have a simple epithelium, but the property is different. So for the PCT, they're going to have cuboidal leaky, you have microvilli. And then the nephron loop, the thin descending arm of nephron loop is going to be simple or tight squamous epithelium. The thick descending arm of nephron loop is going to be cuboidal tight. I should say thick ascending arm. And the DCT is going to be cuboidal tight without microvilli. And the collecting duct is also going to be cuboidal tight. Oh yeah, and the thin is squamous because it does not transport ions. Collecting duct, the two cell types handle different solutes. So Leukovoheny, DCT, and collecting duct play distinct roles in transabsorption of water and ions. They rely on transcellular transport, so they're selective about, I should say, transcellular transport, and they're selective of solutes and water. Leukovoheny is reabsorption for 25%. DCT and collecting duct also absorb 5 to 15% of filtrate. So each Leukovoheny descends into the hypersomatic renal medulla. So, osmotic renal medulla, and then goes back to the cortex via ascending limb. And then remember what we said about this? Juxtamedullary nephrons have loops that extend further into the medulla. And there's a greater osmolarity gradient there, which leads to more water and solute reabsorption. Also, juxtamedullary a vasa recta. This is a special structure. But the big idea is juxtamedullary nephrons. They have a loop of Henle that descends further into the medulla. And in doing so, the medulla has a strong osmolarity gradient. And yeah, that leads to more reabsorption. And the arrangement of water. The arrangement leads to something called countercurrent. So countercurrent is exchanges in different directions. So I write it down. Countercurrent exchange between fluids in different directions. And there's also something called multiplication. Multiplication. So exchange increases as movement continues. And the vasa recta contributes to multiplication. So let's note this down. The thin descending limb, only permeable to solutes. It's only permeable, sorry, to water, but not to solutes. These limbs have aquaporin 1, allowing transmembrane water transport. And as the limb goes deeper, the direction of tubular flow enhances water reabsorption. And the thick one, so the thick ascending, you can pump ions out of the tubular fluid. So this is impermeable to water. So impermeable to water, but can pump ions out of fluid. This is due to nitrogen, potassium, NHCCs. The surface can pump sodium and chloride out of the fluid and interstitial gradient. So it can pump out NaCl, sodium, potassium, chloride channels. Sodium, potassium, chloride channels. And yeah, this is from you can pump it out from the lumen into the ISF. And this is completely impermeable to water, so water is unable to diffuse due to the gradient. An exchange between the limbs and the vasa recta leads to, so vasa recta leads to countercurrent multiplicity. And this leads to exchange between limbs. So imagine like you're pumping out water from this, you are absorbing water here, and then doing so, if you're absorbing water, if you're pumping out water through the aquaporins, then this leads to a more concentration of solutes of Na and Cl in the ascending loop. And the ascending loop is pumping out pumping out water there. So then it's going to be, you're going to have decreased osmolarity. So first, by pumping out water, you're going to have increased osmolarity, more ions. And then here, you're going to have decreasing osmolarity. So it has a multiplicative effect. And and with parallel loops using the vasa recta, it leads to multiplicity. Using parallel loops. And the osmolarity gradient in a deep medulla is partially maintained due to urea permeability in the papillary duct. So within the papillary duct, urea becomes a larger component of tubular fluid, and urea transports are only available in the papillary duct. So the papillary duct would be in more so in the collecting duct. Well, I think the papillary duct would be more so called the collecting duct. So I'm gonna double check that actually. Yeah, papillary ducts are also known as the collecting ducts. So within the collecting ducts, you have urea permeability. So collecting urea. So because urea transports, you're gonna basically be getting more urea into the lumen rather than out of the lumen, from the ISF into the lumen. And this maintains the osmolarity gradient. It's only permeable to urea. This, as a result, urea diffuses out of the interstitial fluid into the peritubular fluid in the deepest parts of medulla. This is, remember, deepest parts of medulla. So in the medulla, in the deepest parts, you're going to have really high osmolarity. And this is going to basically squeeze out any sort of extra water, and then urea is going to pull back in later. And while the urine diffusion also maintains high osmolarity in the deep medulla. I should say that as well. Urea fusion to ISF maintains medulla. And essentially, the DCT and the collecting ducts are also sites for facultative. So for this DCT and collecting duct are for facultative water and solute reabsorption. Remember when we also talked about the uh, the sort of ideas of obligate and facultative water reabsorption? So in the glomerulus and the PCT and the descending, they all have obligatory water reabsorption, but in the ascending, the DCT and the collecting duct, you have more of something where you need hormones to be expressed. Epithelial in the DCT and the collecting duct have little to no ECT and CD, have no paracellular transport, impermeable to water at rest. This is without hormonal signaling, and without hormonal signaling, water will be retained in the DCT and CD even when passing through the high osmolarity. So without hormones, water would be retained in lumen in when passing high osmolarity. And that's not good. So what happens is ADH binds onto both of these, binds onto the DCT and also the CD to add aquaporin 2 on the apical side. This is secreted by the posterior pituitary. Remember that only secretes ADH and vasopressin or oxytocin. And these, by activating these aquaporin 2 receptors, you're allowing the water to finally be reabsorbed. Osmotic reabsorption, and this makes concentration of urine osmotic reabsorption. And this is reabsorbed by the DCT and the collecting duct. And these are the main key effectors for homeostatic regulation of body fluid osmolarity. So the sensor ADH in neurons hypothalamus, they are osmosensitive. So when plasma osmolarity increases, they produce more APs. And the factor effector, the CT and CD cells, facultative water reabsorption via ADH, then decrease in osmolarity. This is example direct feedback loop. And loss of either ADH or uh, like aquaporin receptors, aquaporin 2 receptors, AVPR2. Let me see actually. Oh, so ADH acts onto AVPR2, and this signals for aquaporin 2 receptor. So if you have no ADH or no ADH or no AD, no, sorry, AVPR2 receptors leads to diabetes insipidus. This can be inherited via mutations, which means no AVPR2, or physical damage of epithelium. And in not doing so, you're gonna basically just secrete a lot of urine without, because you like a less concentrated urine, and you're gonna have more urine secretion because you're gonna have too much water. The RAS also regulates iron reabsorption and urine volume, primarily through aldosterone. So aldosterone, remember how we have the RAS signal? It stimulates the NAK pump, ATPase pump, and it secretes NK excretion, NA retention. And this is usually done when there's overall larger volumes of bodily fluids. Reduction in blood volume leads to aldosterone secretion. And by having more aldosterone secretion, you're retaining more water. And returning more water means you're retaining more Na within the, so you're getting, let's say, the lumen, this is the blood vessel, and then this is the nephrons or whatever. So you're getting more Na within it. So aldosterone signaling as basolateral. Remember, this is the apical side, it's basolateral. You're going to have more NAK ATPase channels and as Na leak channels onto apical sides. This happens with DCT. In the DCT, it stimulates the, I'll repeat what I said, NAK channels basolateral, Na leak channels in apical. And this amplifies gradient for Na reabsorption. And the CD side, aldosterone adds ENAC channels, ENAC channels. This is sodium leak channels. This is also enhancing Na reabsorption. So in the DCT, we're seeing the NAK pump regulate ATPase pump in the basolateral side, while we're on the apical side, we see more Na reabsorption using just general reabsorption. On the CD side, you have Na leak channels on its own for reabsorption. And reabsorbing Na, this leads to maintain ion gradients in rest body. Also, retention and reabsorption of water. And this needs to be a homeostasis of fluid in response to blood loss. So let's say we have less blood volume. This activates RAS. The JGA can activate RAS. Angiotensin also stimulates ADH. Oh, I should say angiotensin 2 stimulates H secreting neurons, and then these add on to the DCT collecting duct, also aldosterone, DCT collecting duct for water and ion reabsorption. And yeah, those are the key, and that leads to increased blood volume. More channels. One is actually the night sodium chloride cotransport. This in the DCT allows for reabsorption of Na. We also have Ca absorption, which is regulated by PTH and calcitrol. This in both the DCT and the collecting duct. And you also have acid and nitrogenous compounds, the co-transporters, DCT collecting duct. And lastly, different classes of common diuretic drugs target different transport products along processes along the nephron. So this is labeled as diuretics. This is drugs causing diuresis. Most diuretics are inhibitors for renal transporters. They're prescribed to manage edema or hypertension. So yeah, these diuretics basically just inhibit specific transporters. They allow for more urine production, more urine excretion, I should say. This is done by not retaining the solutes, not retaining water and whatnot. This is all we have, and I'm glad you enjoyed this mini set of lectures I had on urinary system and I guess all anatomy and physiology. And I wish the best of luck to you in terms of your studies. Thank you for listening.